So most of you know what that is. It is a representation, not an actual picture. It's a representation of a SARS coronavirus 2, which produces COVID-19, 19 because it was discovered in 2019. And we're going to talk about viruses, and both why viruses might be considered alive and why they are generally not considered alive particularly. Your typical virus is a casing made sometimes completely out of proteins. In the case of this SARS viruses, it integrates some of the membrane of the previous cell. So it's got a lipid-based membrane. Good news about that is it's more easily disrupted by detergents like soap. And embedded in that membrane are a lot of molecules. The ones that are most obvious are what they call the spike proteins. A virus released from a previous host cell is a tiny, 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 tiny particle. And so it flies through the air, integrated in the case of COVID into respiratory droplets, sometimes in other forms, but they are very, very, very small particles. If they can travel through the really tiny respiratory droplets like measles virus can, then it can hang in the air for literally hours and be breathed in by somebody who could catch measles from that. These guys, nobody knows for sure. It's passed in respiratory droplets, but we don't know what they call the viral load is that a lot of viruses, you need to be exposed to a certain number of actual viruses before they manage to take up shop and cause, and reproduce and spread in you, possibly cause illness, possibly not. For some, it takes a pretty hefty viral load before you can actually wind up with viruses effectively, successfully replicating in your system. With these guys, we don't know. If they're passed in tiny respiratory droplets, maybe they're still viable viruses, but maybe there's not enough of them. If they are in big respiratory droplets, then, which seems to be the most likely passage right now, then those big respiratory droplets don't get very far away from you unless you're spraying or singing or yelling. If you're just talking or you're just breathing, they tend to drop pretty close to you. That's the 6 foot range. Putting a barrier in front of your face catches some of the particles, but also by passing the particles out around the sides of the mask, it tends to drop them much closer to you. So the masks are an effective anti-spreading tool. You probably can't easily pick up the larger respiratory droplets around the edges of the mask as well. So it works. You know, the better the mask, the better it works, but even the masks that people are using will reduce the amount of spread of the virus, both in people that are infected and people that potentially could get infected. These guys, once they're out and about, there is nothing going on in here. There's no metabolism, there's no energy conversion. So that right off the bat is kind of breaking a rule. Even bacteria spreading in spores have active chemistry going on inside them. These guys, the only reaction they have to their environment is if they come in contact with the right molecule on a potential host cell. That's what these spikes are for. It will interact with that molecule, and that will start the process of getting the payload of the virus into a potential host cell. What's inside the virus is code. DNA in a lot of viruses, RNA in a lot of viruses, they're both coding molecules. Coronaviruses carry RNA. Sometimes there's more in there than that. Sometimes there's a protein-based enzyme or a couple of protein-based enzymes. HIV has RNA and 2 very critical enzymes that it needs to get into the host cell to start the process. What's the process? These guys turn a host cell into a virus-making system. In some viruses, they become kind of background workers, where they can take a healthy cell, get it to make and release viruses, and have not a lot of effect on the health of the cell. We carry a lot of viruses, a lot of the Epstein-Barr viruses we carry through our whole lifetimes. And they don't kill our cells, and they don't hurt our cells. They may or may not be associated with certain complications, but they're very effective at doing what they need to do in a host and not seriously debilitating the host. A lot of other viruses, especially a virus that's moved from one host type to another host type, they very often are much less compatible with the workings of the cells and are much more likely to screw the cells up. This virus is very unusual in that it jumped recently from an animal, pangolins, bats by way of pangolins. So they had worked their conditional evolutionary chemistry out on animals with different chemicals than we have. But usually that first jump into humans from animals, these guys are not real good at living in humans, and they tend to cause much worse disease. These guys didn't do that. Also, I'll put in another step. They get in here, they turn your cells into viral making machines. They have codes. The codes are all of the ingredients and the materials needed to make and release viruses from the host cell. These guys have a weird way of doing that, makes them a little unusual in terms of viruses, and that they have RNA. RNA feeds through our protein-making machinery, and so RNA feeds through, and a string of amino acids that's going to become a protein comes out. Well, the first thing that this thing makes is some chemicals that kind of shut down your initial immune response that we typically have to viruses. And you have an enzyme that sits next to the machinery. And instead of making a protein, and then making a protein, and then making a protein, the machinery just continually spews out a string of amino acids. And this other protein goes, oh, okay, that's the end of one of the proteins we need, SNP, and it folds up to become a functional protein. But it keeps going. And then you get the right sort of closing sequence, and it goes, oh, that's the end of another one, SNP, and it forms another. It's like a continuous process with this overseeing protein that snips them apart in order to make all the bits and pieces that a virus needs. That's unusual. There is a company that's trying to go after that particular protein's function, because that's, anytime you're trying to deal with a pathogen, something that gets in you and makes you sick, we would like to poison it. We would preferably like to poison it in a way that poisons it and not you. Viruses use a tremendous amount of our own machinery to make viruses. That machinery is working in a lot of cells that are working perfectly normally. You can't get in there and muck around with that without screwing up a whole bunch of cells. This is why viruses are very difficult to treat. You need them to be doing something unhuman-like, uncell-like, that you can go after. Bacteria have very bacterial chemistry. We can poison that. Antibiotics go after the chemistry that bacteria do that doesn't match anything that we do. Very useful. But with viruses, if you're lucky enough, there's something they're doing that you can actually screw up. With Tamiflu and the treatments for the herpes viruses, there's a particular chemistry that allows the viruses to leave the cells. That's what's being targeted in those. And it doesn't really stop them, but it slows them down quite a bit. It makes it really hard for the offspring to get out of the host cells and spread to other host cells. But that's like the only target, really. We don't know, we don't really have a really good target here. That's why the treatments for this haven't really worked all that well. Most of our treatment targets are targeting our response to it rather than its own particular processes. Okay, so the virus makes more viruses, the viruses leave. Once they're left, they got no metabolism. They've been constructed. They do not grow. They do not develop. They do reproduce, that's one of the things on the list, and they evolve. In fact, they tend to evolve very effectively because they're much more likely to make mistakes in making new codes, so you get a lot more mutants in most types of viral species. And that, so they're going to produce more variety and more potential. There aren't a lot of active mutants in COVID, and the ones that are don't seem to be dangerous in comparison to the other ones. One of them seems to be more contagious, and what they're finding is that very quickly it becomes the typical virus in a population. Its sexual selection, its reproduction capacity is increased. So that's at work. So they reproduce, they evolve. They don't really have typical metabolism. They're not cellular. This one steals membranes, but it's not built like a cell. They do have DNA and protein-based processes. That makes them similar to at least Earth life. They don't respond to the environment in a significant way when they're free in the environment. So you can see there's a bunch of stuff on the list that you can't check off for them, and some pretty significant stuff on the list that you can check off for them. Does this mean they're alive or not alive? Again, it depends on how crazy you are about them having to follow everything on the list in order to get the right label. As I say all the time, it doesn't matter to them whether we think they're alive or not. They're happily doing what they do. They're not capable of being happy, but you understand what I mean. And so that kind of wraps up the first chapter. So we've got the list of what makes things alive. As I mentioned, second semester we'll go through and look at maybe more of the details of those bits and pieces. But here we've got your list, and you've got your viral exceptions and non-exceptions.